All right, so I'm Randy. I'm very short, so I hope you can see me. I feel like I should tiptoe. I'm going to talk about how we get the data and put it into the IDB. So all that data that you're searching, you can see where it comes from and how we organize it. So the scope of the IDB, I think I probably mentioned this, is infectious disease, allergy, autoimmune, and transplant. But I want to say that we do have HIV and cancer data for certain situations. If there was a 3D structure, we capture all data where they show an antibody, MHC molecule, or a TCR in 3D complex with its ligand, regardless of whether that's HIV or cancer. And then also we curate the entirety of a paper. So if the publication included infectious disease epitopes alongside HIV or cancer epitopes, we would capture all of the data. We don't do part of a paper. And then, as Nima mentioned, we have primarily data from the literature, but we also take direct submissions from the submission community. And all of the data in the IDB is experimentally proven. We don't take predictions, and we don't add data that was published previously and just as in a review article. We take the original published experimental data. So this data is very verified in that it was done in an experiment, and then it was published and went through a review process. And we do include negative data and supplemental data. So a lot of data is in the supplemental tables of a paper. We will include those, and as Nima showed on the Immunome browser, you really need the negative data to give you the full picture, so you know it's already been tested and was negative. So all of the experiments showed the binding of adaptive immune receptor to an epitope. So it was a TCR, an antibody, or MHC bounds a peptide or a non-peptidic epitope, but also once we know that a receptor is epitope specific, we capture all of its data. So if a paper describes an epitope-specific antibody, and that antibody also binds to the whole protein in ELISA, it also neutralizes a virus, we capture that data about it too, so you can see everything that that antibody did if it was epitope-specific antibody. And you can search to narrow to just see the results that you're interested in, pick the epitope, and then pick what antigen you wanted that experiment to be about. Because we get this data from all of the literature and we have a team of curators, we had to have some structured rules as to how we were going to enter the data regardless of what paper it came in and what author published it. So we decided to define an epitope if it's a peptide as 50 amino acids or less. So if we know that the MHC isn't binding 50 amino acids, the antibody isn't recognizing 50 amino acids, but that was a way to cut down what we would put into the IDB. So if they test overlapping 50 mers, we capture that. And if another author goes on to define precisely the epitope, we also capture that. So when you look at that Immunome browser, you're seeing the entirety of all the peptides that all different authors tested and published over time. So very often, originally, someone tests a large peptide first, and then they further narrow it. So we curate both. And anything 50 amino acids or less that's tested as immunogen or antigen. So they may not claim it was an epitope, but if they vaccinated with 50 amino acids and tested for an ELISA to a whole protein, we would capture that data because it's still useful to help you get to where the epitope really is. If that was where they left it and that was the only publication, it still helps you a little bit. So we put that in. And then the discontinuous residues, when they're shown either by 3D structure or point mutation, we capture that, and we would curate precisely the peptides known to be contacting for the antibody or MHC. And then non-peptidic epitopes, we have that data too. These are like carbohydrates, lipids, chemicals, drugs. They just need to be smaller than 5,000 Daltons, tested as immunogen or antigen, we capture that. And then across all the papers, we have to have a minimal amount of information to think that it's usable enough to be entered. So if we can't tell what the host was, or we don't have the precise amino acid sequence, if they just give us positions in a protein, we need to know exactly what amino acids, because proteins can be highly variable. So we need a minimum amount of information, and we contact authors if this is not available in the publication. We'll try to get out of them what was the exact peptide you tested, or if it's an antibody and you don't know what species it came out of, we wouldn't capture it, but we will write and ask. And we'll look at cited references, because sometimes that information was just buried in a different paper. Nima showed the top part of this graph before of where we get the data. It comes from primarily from PubMed. We go back to PubMed every 2 weeks and do a query, and we get all the data and run it to the classifier, and then it's manually reviewed by Alex, as Nima mentioned. And then of the 230,000 papers that we retrieved this way, approximately only 20,000 so far have been deemed curatable, and that's because they have to be relevant to our scope and contain all that minimum criteria. And they can't be a review article. We're just showing previous data. So once that data is highly vetted and put in by our team of curators, and then reviewed by our team of curators, it gets to the website, and that's the data we're giving to you. And as Nima mentioned also, how that data falls, I already forgot I'm supposed to push, into the different categories, totally just reflects the literature. This is based on what people are publishing. If people published more epitopes in a different area, the numbers would change. So we're agnostic, we just take what's out there. So we want to make sure the data is consistent and accurate that we give it when we give it to you. So we have a lot of measures that help keep us consistent across different curators and different authors and different publications, because there's a lot of variety of how people publish. So what we do is we have our team of PhD level curators who understand these experiments and know how to read these papers. They read them and enter the data. And then a second curator reviews everything that they did, and they work together to make sure it's complete. And then we have formal curation guidelines and a peer review process. And you can access our curation guidelines through a wiki page that I'll show later, and you can also find it on the website. And then to design the database in the first place, and if we're going to add new fields or change something, we go to external immunological experts. I mean, Bjorn and Alex themselves are immunological experts, but we don't want to be insular. We want to reach out, and that's why we have workshops to talk to people. We go to booths to talk to people. And we want to, if something is a specific niche area, we will go to the current immunological experts in that field and work with them, because we want to serve you the best we can. And then there's built-in validation in the curation system itself. So as the curator's entering data, that controls what data they can enter down the line. So if they enter that there's a disease, they're required to enter what was the stage of that disease. But they can't enter a disease stage if they hadn't entered a disease. So there's all this interfield validation. And we also limit the type of data they can enter, and I'm going to show a few slides on that. So the submission community is these are the people that we take data from directly that aren't necessarily in publications, but they might be. So they primarily, to date, have been epitope discovery contract holders, where they received a NIH contract that required them to submit the data. And they span all sorts of different types of epitopes, mostly infectious disease, but also allergy and autoimmunity. And we'll take submissions from anyone, so if anyone has data that they're interested in submitting, Nima is the contact person for that. And this process is very similar to the curation process. They submit the files rather than the curator entering it, and there's two ways to submit. You can do one by one using a DST wizard that helps show you how to submit, but most people are submitting a large amount of data, and that's why they're submitting it, because they're going to publish the 20 best epitopes, but they actually tested 3,000 epitopes, and they want to get that data out there so other people can use it, which we're very grateful for. So we have a spreadsheet template that they enter the data into, and then it's uploaded to the site, and it undergoes the same validation as the curated data. The same in-house validation, but also human. Nima or another curator will review all of that data and make sure it looks correct and accurate. And then we hold on to it until they tell us to release it, because very often they're planning on having it released alongside their publication. They want their publication to come out at the same time. And you can submit ahead of time, get a submission number, and put that into your publication, so that it refers back to the IDB, and they can see the rest of your data in the IDB. The colors are jarring. It looks different at home, Sheridan. So basically, the IDB is that we start with the reference, and each reference, to be relevant to us, has to have data about an epitope. It can have data about 1,000 epitopes or a single epitope, and we will take it. And each of those epitopes must have been tested in one or more experimental assays. And the structure of the assays, you'll see again and again, and they have all these fields that we capture, up to 400 fields per assay, but they're primarily about the host, whose immune response was being studied, what was their immunization process, did they have disease, were they injected with something, and it's either antibody, T-cell, or MHC assay, and then what was the antigen actually tested in that assay. And that means you could have had diabetes and taking serum out of those donors and done an ELISA with peptides or proteins or something. And then to structure the data in the IDB, all of those different 400 fields, we like to control them and keep them accurate and use proper nomenclature. And we do this using ontologies, and that I'll go into detail about what that is, but it gives us a standardization for how terms are entered in the IDB. So each ontology has its own field of experts that determine the proper nomenclature for that type of term. So there's NCBI taxonomy, determines the names for species, and the MHC ontology determines the proper nomenclature for MHC molecules, which is really complicated. And it also provides a hierarchy, a relationship between the terms that helps organize the data, that makes the search easier, and also makes it easier for our curators to enter the data because they're picking from these ontology terms rather than typing anything in. We hope that it facilitates searching and enhances your experience, and we'll show you today several ways that we think it does. But also, it makes our data interoperable with any other resource that uses those same ontologies, because they're storing their data the same standardized way, using the same terms and the same identifiers. So then if you have another resource you're interested in, they're storing the data the same way, you can combine our resources. So this is one example. This is the NCBI taxonomy. We use this to organize how we talk about the organisms epitopes are derived from. So most of our epitopes are derived from proteins, and those proteins are derived from an organism. And organisms have high level, like all viruses or all bacteria, but we also have very specific viruses and all of the strains that may have been studied for that virus. So our NCBI tree, when you see it, it's limited to those species for which we have data. So you can also, just by navigating that tree, see which species have been studied and which particular strains have been studied. By using the NCBI taxonomy, we get all these synonyms, and this makes the search much easier because you can type a really short synonym, or you can type just part of a strain, rather than typing out the full name of a virus, which is tedious. And you can get to it, and we also provide links back to NCBI so you can see where we got that identifier from. And then this is proteins. So there's lots of proteins in GenBank and in UniProt and PDB. And we want to organize those for you because just searching on a protein name, you may or may not get the protein you're looking for, or you may miss a lot of data for that same protein that has a slightly different name. So instead, we provide this information using UniProt reference proteomes. So UniProt takes for this species, these are all the proteins that that species has. So we take that package set, and we show you the proteins from that species for which we have data. So each of these proteins from the reference proteome from UniProt, it's representative of all the matrix protein data we have for that species. And again, we get the synonyms when they have them. And then we do this also for chemicals. We organize them using the Kebi ontology, and we organize, this is just about chemicals. So from using Kebi, we get all these synonyms again that we may not have the expertise for, and link outs to other resources that also use the same thing. And the structures, which amino acid structure is simple, so you can see that clearly on the screen, but to get to, if you really want Smiles information or something, you can link through the Kebi ID to get that. So again, for the host of the experiments, so it may be human, but it may also be cow or camel or cat or a particular strain of mouse. We use NCBI taxonomy again for that. And then we use the disease ontology for diseases. And by using the disease ontology, we get a structured tree that organizes diseases, but we also get a lot of synonyms and link out to other resources that they link to. And then for assays being one of our most important features, that's how we organize a lot of the data. We use OBI, the ontology for biomedical investigations. And so it is about experiments. And so they provide us our tree structure for the experiments. And this lets you search for all T-cell assays, for example, or all T-cell assays where a cytokine was released, or a specific cytokine. If you want to just look at IL-2 assays, you don't care how they were done, you can search using the tree. But also for our curators, the precise platform that was used to measure that cytokine, the curators can pick. So it makes it easy both for curation and users. And then for MHC restriction, we use the MHC restriction ontology, MRO, and this provides the relationships between all the chains that make up an MHC molecule. So you could search for all MHCs containing a particular alpha chain or a particular beta chain or a particular combination, and you get that level in that organization because of the ontology, so you can search for all class 1 molecules, all human class 1 molecules, or a precise or a vague query based on what you need. Then I think it's even mentioned too, the power of the IDB is to put all this data together in one place. So by representing all the epitopes out of all the papers, you can then see the accumulation of all of the work that's been done. So there may be a lot of studies on one epitope, or there may only be one, or they may build and always be positive for the same thing, or they may be different among papers, and this is because of all the detail. So a particular epitope may have been positive in an ELISA for a donor that had diabetes and negative in a healthy rat. So you have to see all the contexts, and that's why you really need to know how to query the database so you can get to just seeing the outcomes that are relevant to what you're particularly doing. And I think, is that the last one? Okay, now are there any questions? And we're going to go into more detail about everything in the afternoon.
Yeah, so I understand everything, and I think it's just terrific. But my question is, why not go also to the patents many scientists have, where they really have epitopes that go towards diagnostic and vaccines, but they haven't published precisely because they don't want their intellectual property to be revealed until the patent comes through, or even if the patent has come through. So why not use patent information as well? Okay,
so the question was, why don't we use patent information? And the answer is that we actually do. So at the beginning, we focused and made sure that we got patent information. There hasn't always been that much of it, but we do still get submissions from time to time that our patent is patent information, epitopes that are used in a vaccine. We will capture that. Or if a publication refers to a patent, we will take the time to look up and see if we can access that patent. And Alex has more to say about it.
Is this on? Yeah, so certainly patent applications are a source of information. The challenge with patent application is that that is not peer reviewed. And actually in a previous life, I worked on a company that developed epitope-based vaccines for cancers, and the patent lawyer will do their best to confuse and put all sort of information in there. Some of it is prophetic. It's very hard to tell what is real and what is trying to, which is their job, but they're trying to cover as much possible. And it's, it gets into a little bit of a shady area. And so in the start, we looked at it, and it was really hard. And at the end of the day, also, as Randy was saying, the good stuff eventually gets published because the patent gets published, and so.
A lot of times in the publications, they won't put the epitope sequence in the publication, but they'll refer to the patent. So we'll use the patent as the source of the epitope sequence.